Hi, in this video I'm going to talk about the gene, genome, and the different type of the nuclear DNA sequences in the human genome. I hope you enjoy watching this video. If you like it, please subscribe the channel and click on the bell for receiving the upcoming videos. Although there are 5 different definitions for the gene, but the concept of all of them is approximately the same. Now I'll review all these 5 definitions and at last I'll summarize that into a simple sentence. In the first one, genes defined as observable effects on live organisms that can be transferred from a generation to another. This is the offered definition of Mendel and the first geneticist. In the second one, gene is the observable diversity that results in a certain clinical disorder. This is the view of the medical geneticist. Two remaining definitions are in general. In the first general definition, gene is a special DNA sequence which provides a functional product. This product can be a polypeptide or a functional RNA. In the second one, gene is the functional unit of the genetic information which is encoded in the genomic DNA. And in the third one, gene is the continual DNA sequence that encodes a protein. As I said before, all these definitions nearly have the same concept. So in summary, we can say that the gene is the DNA sequence that is coding a polypeptide or a functional RNA. In detail, we have 20,000 to 25,000 polypeptide or we can say protein coding genes and also 20,000 to 25,000 functional RNA coding genes. Overall, we have 20,000 to 50,000 genes in our genome. The structure of most genes consists of the exons, the introns, 5' and 3' untranslated region or we can say 5' and 3' UTR in abbreviation. Why is it most genes? Because exceptionally the SO genes don't have the introns. The exons are the most part of the genes that are transcribed to the RNA, and finally they are translated to the proteins. Thus they determine the protein function and structure. The introns, they are located between the exons and also be transcribed but omitted in mRNA splicing, so they aren't translated to the protein. 5' and 3' UTR, they are transcribed, present in the mature RNA but not translated to the protein. There is a difference between 5' and 3' UTR and the introns, and that is the 5' and 3' UTR are presented in the mature RNA, are not omitted in the mRNA splicing, but the introns are. 5' UTR located in the upstream of the coding region, I mean exons and introns, and 3' UTR located in the downstream of the exons and introns. As you can see in this figure, the 5' UTR are exactly located on the upstream of the exons and introns, and 3' UTR are located in the downstreams of them. Now you may be wondering what a genome is. The genome is a term that is a combination of the word genome, this is a German word that means genes, and the word this is adapted from the chromosome. Genome refers to all the DNA material of the organism, including both the genes, I mean coding DNA, and the junk DNA, I mean non-coding DNA. I'll discuss this afterwards. Mitochondrial DNA and the chloroplast DNA. Chloroplast DNA presents in plants, not in animals. The DNA is composed of two antiparallel strands in the form of the double helix. This figure shows the double helix form of the DNA. But the question is, what in each strand of the DNA is? We can see this issue in two aspects. The first one is the structure, and the second one is the sequence. In terms of structure, each strand is made by an abundant, maybe 1,000 nucleotide. Maybe I dedicate a video to this topic in the near future. And in terms of sequence, the genes and the junk DNA are the two major categories of the DNA sequences. And also, there are some classes that I explained that in the subsequent table. You can see the gene and junk DNA in this figure as well as the gene structure as I said before. In this table, you can see the classes of the genes and the junk DNA in our genome. Only the genes are transcribed, whose classes are protein coding genes, or we can say polypeptide coding genes, like collagen genes that produce collagen protein. Long non-coding RNA genes, or we can say LNC RNA genes, like microRNA genes. The function of microRNA is in regulating the gene expression. Tandemly repeated genes is another class, like UDS and RNA genes and rRNA genes. The function of UDS and RNA is in mRNA splicing, and RNA is the component of ribosome structure that translates the mRNA to the protein. Protein coding genes includes 40% of our genome, but there is a point in this value. This value takes into account exons and introns. If you only consider the exons, the protein coding gene nearly consists of 1.5 to 2% of their genome. nscRNA genes consist of 15% of our genome, and tandemly repeated genes includes a small part of our genome that you can see the exact number in this table. The junk DNA isn't transcribed and probably involved in regulating the gene expression. This also consists of the repetitious DNA and intergenic regions. The repetitious DNA include simple sequence DNA and interspersed repeats, or we can say mobile DNA elements. The simple sequence DNA contains 6% of our genome and are divided to major satellite, mini satellite, and microsatellite. The major satellites are located in centromeres of the chromosomes. Mini satellites are located in the telomeres or are scattered throughout the genome, and microsatellites are scattered throughout the genome. There is a difference between the satellites, and this is in the length. Interspersed repeats include DNA transposons, LTR retrotransposons, non-LTR retrotransposons, and processed pseudogenes. DNA transposon contains 3% of our genome, and they are the transposomes that are transported throughout the genome via DNA mediator. LTR retrotransposons contain 8% of our genome, and they are the retrotransposomes that are transported throughout the genome via RNA mediator, and also has the LTR, or we can say long tandem repeat, in their structure. Non-LTR retrotransposome, they are the retrotransposome without the LTR sequence in their structure and are divided to LINE, or we can say line. This is the abbreviation of long interspersed nuclear elements, and SINE, or we can say SINE. This is the abbreviation of short interspersed nuclear elements. Lines contain 21% of our genome, and the length is more than 100,000 base pair, and the SINE contain 13% of our genome, and the length is 300 base pair. Processed pseudogenes. They are the pseudogenes. They are the result of the reverse transcriptase enzyme function. As you know, the pseudogenes are really similar to the coding genes, but without the functional expression. And finally, intergenic regions. They contain 25% of our genome, and they are the spacer between the coding genes. This question may arise for you. If I sum the fraction of the human genome, the result is more than 100%. The answer is because interspersed repeats are counted twice, once to show the different classes of human mobile DNA elements, and second, as part of the intergenic regions and protein coding genes, where they are located in intros and 3' untranslated regions of terminal exons. For more information and details, you can see these books: Emery's Elements of Medical Genetics 15th edition, Thomson and Thornton Genetic Medicine 8th edition, and Molecular Cell Biology 8th edition. Thank you for watching this video.